Greetings and welcome back to Old Ways Rising Farm, where we are going to join together in a conversation about legumes and nitrogen fixation. Nitrogen fixation is a process where plants buddy up with some bacteria to take atmospheric nitrogen, a form they cannot use, and convert it into forms of nitrogen which they can use, producing their own fertilizer in essence. Now, many gardeners have the idea that beans and peas are good for the soil, and this is why. But there's a lot more to the story than your garden beans and peas, and a lot more even than alfalfa and trefoil, and bird's foot trefoil. So let's take some time to discuss the less often mentioned legumes, such as this showy tick trefoil that's just being absolutely beautiful today, as well as other groups of plants that fix nitrogen by other means, and some legumes where there's a question mark about how much nitrogen they fix. Let's go dig in. Here's a couple more examples of some often overlooked classic nitrogen-fixing legumes. This is a lead plant. There's 3 of them in there. Not the best germination in that pot. But these are a lovely tallgrass prairie species that is a great nitrogen fixer and has a lot of use as a potential fodder plant that's been understudied and underdeveloped. The same can be said of this little fellow. This is a beautiful, beautiful plant when in bloom. This is Baptisia australis, the blue false indigo. Again, lots of potential as a fodder plant. It's been overlooked and understudied, and oh my gosh, these guys are drop dead gorgeous when they're in full bloom. This one's fascinating for another reason, Tephrosia virginiana, the Virginia goat's rue. This is an Appalachian endemic species, and it is the producer of rotenone, the natural insecticide rotenone, so you can grow your own insecticide with this one. Its close relative, Tephrosia purpurea from the Pacific Rim, is also extremely fascinating. It's super important in Polynesian aquaculture because it produces chemicals which will stun fish but not kill them. Rotenone just flat out kills them. Don't use this guy in aquaculture. Bad news bears. Right? But again, beautiful, beautiful purple and white flowers when it's in full bloom. Great nitrogen fixer, and you can grow your own spray materials if you need them. This one is another really interesting Appalachian endemic. This one is a very restricted natural territory, so buy seed responsibly. It's a close relative of the Baptisias, and it's got a yellow flower. Again, spectacularly beautiful, showy yellow flowers. This is the Appalachian buckbean. And then another yellow flower species, in this case from Europe. This is the golden chain tree. Okay. The small tree, it's a small tree or a large shrub. This will be planted in around the edges of windbreaks and around apple trees and things like that. Because it's a short tree, it won't overgrow the short fruit producing trees. So it's a great companion plant for your fruit trees. So these are just some examples of some legumes. What they all have in common is that they have a very complex interaction with bacteria in the family Rhizobiaceae. These bacteria are free-living soil bacteria. They're very, very common. They have a flagellum when they're in the soil, so they can wiggle around the interstitial water in between the soil particles. When they come up against the root of one of these guys, they will exude a hormone that a signal molecule. It's not the same as hormones in animals, but we'll call it a hormone for this video because that's a relatable term. They'll exude this hormone, and it basically says, knock, knock, I'm here, can I come in? The plant root will sense this hormone and produce its own hormone, which says, sure, you're welcome, come on in, fix nitrogen for me. Once inside, the bacteria will shed its flagellum and lose the ability to live independently in the soil. It will then say, hey, I'm in here, I'm dependent on you, again through another series of hormones. The plant will respond to this by producing a nodule on the root and filling it with sugars to supply the bacterium with energy. So the plant has its leaves and is fixing carbon producing sugar, which it feeds into the nodule. The bacterium then takes these sugars and uses them to convert atmospheric nitrogen into a form that the plant can use, which takes copious amounts of energy, thus requiring the sugar from the plant. Let's talk a little bit more about the details of how this interaction works. Okay, let's take a couple minutes here inside and discuss some of the scientific details pertaining to the Rhizobium bacteria legume nitrogen-fixing association. While we were outside, we discussed this basic idea of free-living, flagellated swimming bacterium exchanges some chemical signals with the plant, asking to come in. The plant exchanges some chemical signals back, inviting it to come in, at which point the bacterium changes its chemical signaling pathway to cause the plant to produce a spongy growth we call a root nodule. What we need to do is to expand on this and talk about what bacteria are involved and what they're actually doing inside of this root nodule. So let's start with the bacteria. While we're discussing legumes, we're discussing the family Rhizobiaceae. Quite a few genera in that family, but we're really interested in one clade, which is a group of closely related in evolutionary terms genera. Okay? So within this clade, there's quite a few of agricultural interest. First are the Agrobacterium. They're bad guys. These are a common agricultural pests causing a disease called crown gall. You may have seen this if you've been walking in the eastern forest. Look at hemlock trees. Look at black cherry trees. They both very commonly have crown galls on them, and you'll have this nice straight tree, but then there's this big swelling on the side. That's a crown gall infection. The Agrobacterium, they enter through a wound in the stem of the plant being discussed, and they turn on the same, actually it's literally the same chemical signaling pathway that the good ones use, but now they're forcing the tree to feed them and they're not really giving anything back, so it's a parasitism. Most of the ones in forest trees aren't that harmful. They just change how the tree looks and make burls that woodworkers enjoy playing with. But the ones that grow on certain other trees, peach trees can get really severe crown gall infections. Grapevines can get really severe crown gall infections that do a lot of harm to the plant. So they can be benign or they can be very, very harmful depending on the species being discussed. So those bad guys, the genus Rhizobium and the genus Rhizobium, most of them are nitrogen fixers. Some of them also cause crown gall. So these are mixed. And then Pararhizobium, Neorhizobium, and the genus Ensifer, they only fix nitrogen. They don't have any baddies in them. Within this clade, probably the most basal and ancestral is the genus Rhizobium itself, which does a little bit of both. And then later on, the Agrobacterium specialized in causing crown gall, and the rest of these really specialized in only fixing nitrogen. When I first studied this, I kind of learned it in terms of Agrobacterium and this crown gall behavior being the ancestral trait, which then later evolved into nitrogen fixing. But phylogenetic trees I've seen more recently are making me question that a little bit. But that still could be the story. You could just have the crown gall causing Rhizobium being the ancestor. So it's a little bit more complicated than when I saw this for the first time as a student on a whiteboard. But that's normal. Science is continually adding new information to the mix, and we're going to see where some more of that comes in with some other plants here in a few minutes. That's okay. That's good. That's healthy. Now, once we have a bacterium of the proper genus and species to associate with a legume of the proper genus and species, they've exchanged their chemical signals, the bacteria are on in the root nodule. What are they going to do? Well, we need to talk about this group of enzymes, the nitrogenase enzymes. Okay, there's quite a few members of this family of enzymes. You have different ones in different species which work similarly, but with some differences in the details. Okay? What they're going to do in general terms is they're going to start with atmospheric nitrogen, N2 gas. That's going to dissolve and mix in with the liquids in the cell. You're going to have some hydrogen ions associated with various organic acids, as well as some membrane properties in the cell. And then you're going to take some energy. We have to apply energy in order to take this nitrogen in its most stable form and convert it to the much less stable ionic form, which is then useful to the plant. So we need to add energy to the system to do that. The energy capsules, or you can think of them as like batteries by analogy. Don't take that analogy too literally, but it'll work for this discussion. That a cell uses is what's called ATP, adenosine triphosphate. Okay? In this case, they use ATP in a form which is interacting with a magnesium ion. Again, magnesium is one of these micronutrients that we talk about needing to add when we fertilize a garden bed. Here you can see where it's actually playing in part of a biochemical function. This is what all of your plant nutrients do. They are raw materials that plant cells use to make stuff, or in this case, bacteria cell is using to make stuff and to do stuff. They are very real building blocks of cells. So here we have our magnesium micronutrient and our ATP forming up. In the first pathway I drew, we have 16 little batteries being used. In the second, we have 40 little batteries being used. So this consumes quite a quantity of energy. Most biochemical processes, we're not going to see this many ATPs. It's actually kind of an oddity, right? So this is very energetically expensive for the plant cell, which is why, for the bacteria cell, which is why it needs to be up here in a root nodule being fed by the plant cell adjacent to it. The plant cell can make a lot of sugar because it's because it photosynthesizes, as I stumble over my word, and feed that sugar down to these bacteria, which then the bacteria can convert into ATP and use that energy supply to build on this nitrogen. Now, all of these ingredients get into position to interact with these enzymes, and the enzyme does its thing. We're not going to talk about the details of the thing. We're going to talk about what comes out. So in one pathway we get NH3, ammonia. In the other we get ammonium ions directly. Now in water, this very quickly converts to that. So even though it spits something different off of the enzyme, it's going to end up down here as ammonium. Ammonium hydroxide is what's going to go into the liquid of the bacterial cell and then be fed to the plant cell. You also get out some hydrogen gas, H2, and you get out 16 magnesium-bound adenosine diphosphate. So what happened here with our battery? Well, in its fully charged state, it has 3 phosphates. In its depleted state, it has 2 phosphates, and this is the phosphate ion that we're talking about. And again, this is the phosphate ion that you're adding when you dump fertilizer onto your garden. Okay, that's this raw material, and here you can see where it's playing an important biological role. So, understanding this is important. Nitrogen gas is never in short supply. It is the most abundant gaseous element in our atmosphere. Okay? But the bacteria in and of itself is going to struggle to make enough energy in and of itself to do this in large abundance, to actually have enough ammonium going into the system to fertilize that soil and build it up and perform their ecosystem service of being an early colonizer of degraded sites. But when we have the bacterium in the root nodule as part of the plant can fix that nitrogen. The plant can fix that carbon, allowing the bacteria to fix the nitrogen, which allows the plant to colonize a degraded site it would not otherwise be able to colonize. So this is the benefit of the legume rhizobium mutualistic symbiotic relationship. But there's a little more to this story too. it turns out there's some legumes that don't form root nodules. And it turns out there's some discussion about them. So let's go look at a couple and talk about that part of the story. So these are a few of the non-nodulating legumes. They do not produce any nodules whatsoever. And this is where you kind of get into an active argument and question mark in the botanical community about what to say about these things vis-a-vis nitrogen fixation, right? So in the early studies of the nitrogen-fixing symbiosis, it was just what I had up on that whiteboard a minute ago, right? You have the nitrogen-fixing diagnostic nodule, and if you don't see that, you don't have a nitrogen fixation. And this is totally reasonable in the early years of the study of this phenomenon. And a lot of the folks that were studying and writing about this and using nitrogen fixation as an evolutionary case study, like Dr. Raven, are some of the key botanists who wrote all of the textbooks. So this was written into all of the textbooks. But it's complicated. The groups of non-nodulating legumes are distributed widely across the legume family. And if you follow the phylogeny of the legume family and you assume that non-nodulating legumes are non-nitrogen fixing, then you have to conclude that nitrogen fixing evolved 3 separate times in legumes. That is not very parsimonious. Okay? Occam would be very, very sad by this. But there's another set of studies which look at the non-nodulating legumes, and they look at the nitrogen composition in the leaves, and they say, it's pretty much identical to nodulating legumes. Isn't that interesting? So they looked at the roots, and they did some biochemical testing, and the genus Cercis, to which this redbud belongs, is one of the genera which is regularly described as not nitrogen fixing based on the lack of nodules. But when they did this biochemical test on the roots of one of the Eurasian species of Cercis, they found nitrogenase activity. So, wrong. Cercis is a nitrogen fixer based on the biochemical testing. Now, only a few of the non-nodulating legumes have been really studied in this way. These haven't, right? This is a Kentucky coffee tree. This is honey locust, and that is American yellowwood. Okay? So these are all legumes, but they are not nodulating legumes. What can we say about them? Well, right now, we can say definitively that they need more study. But the studies on the biochemistry of the roots that show nitrogenase activity also section the root into the little plant histology and observed bacteria clusters in the root which look pretty much identical to the bacteria clusters in nodules in the other legumes. So, the preponderance of the evidence is pointing in a different direction now. That these non-nodulating legumes probably represent an early, primitive, if you will, example of the nitrogen-fixing symbiosis that isn't fully fledged, fully formed to produce the root nodule. So if we base our assumptions on that, then we can say that the evolution of nitrogen fixation in legumes happened once. We can return to a point of saying all legumes are nitrogen fixers then, or probably nitrogen fixers. You can always have reverse mutations, weird stuff go on now and then. But we can say that the evolution happened once, and that from that point the evolution of producing nodules happened thrice. That's a much more parsimonious consideration, especially when you remember, as I discussed on the whiteboard a few minutes ago, that the nitrogen-fixing bacteria are closely related to the crown gall bacteria. So that ability to produce aberrant plant growth is there in the family from the get-go, right? So that is not nearly as weird as saying nitrogen-fixing evolved 3 separate times. So, In my garden, I'm going to make the assumption that these guys are all nitrogen fixers until proven otherwise. Because that is the parsimonious assumption. That doesn't mean it's right, but it is the parsimonious assumption. And these are all great plants for low quality soil. So, regardless, even if that assumption is wrong, you're still not doing any harm. Go for it, plant what you like. Now, there's nitrogen fixing beyond just the legumes. Let's look at a couple other plants. This little beauty has the common name sweet fern. Comptonia peregrina is the Latin name. It's another lovely little Appalachian endemic. And it grows on very, very dry sites, dry hillsides, where it doesn't get a lot of shade from taller woody vegetation. Because it's a low-growing 3-foot tall by 4 or 5 foot wide mounding plant, and it needs full burning sun. So it cannot compete with tall forest vegetation on moist-rich sites. It's related to the rest of the Myricas. It's in the Myricaaceae family, the wax myrtle, the sweet gale, a lot of those classic sand dune plants. And this family, all of them are nitrogen fixers, but they're not even closely related to the legumes. Well, how's that work? Let's go back up to the whiteboard and we'll talk about these plants for a minute. So in order to explain some of this broader context of nitrogen fixation, we need to zoom out a little bit. We need to look beyond just the legumes and beyond just nodulating bacteria and talk about a couple other things. In order to delve deeper, we need to throw a couple more technical terms out. The first is Diazotroph. That is a generic term for any bacterium which has some sort of nitrogenase or nitrogen fixing capacity. Okay? Turns out there's a lot of them. Many of these are endophytes. Second important term. That just means they're inside a plant, right? So everything we've been talking about in root nodules up to this point is definitionally an endophyte. But pretty much as far as we know, all plants have endophytes. They don't all have root nodules. Okay, so there's a lot more to endophytic behavior than just root nodules. That's going to get real important here. But the root nodules are an important example of endophytic activity, specifically endophytic Diazotrophs. Okay? The next most studied group of endophytic Diazotrophs that do form root nodules, and hence they're well studied and were noticed early, are Frankia bacteria. Okay? This is a genus of bacteria. Another term for them is Actinorhizae, and plants that associate with them are thus often called actinorhizal plants. Okay? Now, Actinorhizae also include dental bacteria. We're going to come back to that in a second. But first, the plants. Some well-known groups of actinorhizal plants are the genus Ulmus, which are the alders. This is a genus within the Betulaceae, the birch family. The genera Persia and Dryas in the rose family, Rosaceae. The whole family Casuarinaceae, which are the Australian pines, very commonly planted in the dry tropics. If you haven't been to the dry tropics, you may never have met one of them. Google it. They're very easy to find. Very common plants. And Rhamnaceae, these are the buckthorns. Alegnaceae, see buckthorn, autumn olive, and their relatives, the Alegnaceae. And the Myricaaceae, the wax myrtles and the sweet ferns that I used to introduce this idea. Okay? So those are all examples of Frankia associating plants. These are, again, specific associations, and they do form a characteristic root nodule, very similar to what we see with Rhizobium. But not all Diazotrophs are endophytic. We also have the blue-green algae. So pond scum is fixing nitrogen. Just is. They don't have to be associated with any other plant. They're just nitrogen fixers. And blue-green algae are present in a lot more than just a scummy farm pond. This is a soil sample that I was working with and showed in an earlier video on soil horizons, and I forgot to clean it up, and now I'm happy that I did, because look at this nice little clump of blue-green algae floating around there in the middle of this water. That came from the soil. That's nitrogen fixers from this soil, not associated with a plant. So there's more to it than just endophytic nitrogen fixation. Blue-green algae do have endophytic relationships, though. Okay? We have, in addition to the free-living ones, we have some that associate with a zola fern, which is a floating fern, very important in Polynesian agriculture. We have blue-green algae associate with some cycads, and we have blue-green algae associate with some bryophytes, like liverworts and hornworts, as well as lichens, which are half blue-green algae, the other half being fungal. Okay? All of these are nitrogen fixers. And yes, even lichens do contribute to the environment, because as the rain washes down the trunks, it dissolves some of the nitrogen that they fix and washes it to the tree roots. So they are an important part of forest ecosystems. Now, let's go into the weird ones. There is such a thing as nitrogen fixation in animals. Termites and shipworms both have nitrogen fixation in their gut flora. They have Diazotrophs that have been identified in their gut microflora. How important is this to ecosystem function? We don't know, but it's there. Also, remember I mentioned that Actinorrhizae are closely related to dental plaque bacteria? Well, guess what? Your teeth are fixed in nitrogen. Every day, you swallow about 1 hot dog worth of nitrates that are produced by those dental plaque bacteria. So you're a nitrogen fixer. Pat yourself on the shoulder. It's a good day. Now, there's also many soil bacterium which are important in various communities as well as other biogeochemical processes that include Diazotrophs. Okay? So a well-known example is Azospirillum. This is a common rhizosphere bacteria. The rhizosphere is just a technical term for the root zone. So rhizosphere bacteria are bacteria that will find a plant root and live right up next to it. The plant root produces exudates, sugars, growth stimulants, sometimes surfactants, which help stuff dissolve in the soil, that promote the growth of these rhizosphere bacteria. And the plants will modulate those root exudates to attract the most beneficial ones. Okay? So it is a form of symbiosis, but it's not quite as explicit as true endophytes. They also tend to be very non-specific. So Azospirillum are happy to live in the rhizosphere of any old plant. So now just because you have a plant, you're promoting nitrogen fixation. Okay? And then when we get into the deep levels of the soil, into the anaerobic regions, like the low at the bottom of the sea horizon, we start to get into things like sulfate-reducing bacteria, Clostridium bacteria. That is a pathogen, but it's also an important soil bacteria. It's ubiquitous. It's all over the place. Methanococcus and others. These are just a couple examples of a very large group of soil bacterium. They're responsible for hardpan formation, caliche formation, a lot of biocementation, a lot of really interesting biogeochemical processes that we're not going to talk about in this video, but many of them are Diazotrophs. What is the importance of this plant unassociated nitrogen fixation in the soil horizons? We don't know yet. Has not been fully defined. How important is this to the health of your forest and your garden? We just don't know yet. But from this board, I hope you're seeing just how much there is to learn and to talk about. And I'd like to take just a couple more moments with you and share some really recent discoveries. This little plant friend is Paulownia fargaceae, not Paulownia tomentosa, the invasive species. If anybody is worried, no, I'm not planting invasive species here. But Paulownias are a fascinating tree with a lot of potential for permacultural settings. And they're going to be a example, a case study in the current research and where the research is going. There's an old story about an old gentleman who lost his wedding ring and was looking for it under a street lamp. And a police officer comes up to him and says, looks at him worriedly, studying the ground, and is worried about this gentleman and says, "What's wrong, sir? Can I help you?" He said, "I lost my wedding ring." The police officer said, "You lost it right here?" He said, "No, I lost it over there." And the police officer said to the gentleman, "Well, then why are you looking here?" And his answer was, "Because this is where the street light is." Okay? That's been the hang-up with nitrogen fixation and the study thereof for a long time. We've known for a very long time about root nodules. We've known how to recognize them. We've known how to look for them. That was the street lamp. Okay? So for a long time, all of the studies about nitrogen fixation were very, very heavily biased in favor of nodulating species, and precious little else was done. Now, in more recent time, we've developed a tremendous number of tools for studying biochemistry in very new ways. We have mass spectrometers that can do de novo protein sequencing very, very quickly and let us look at just enzyme activity across a large landscape very rapidly. We have tools that can allow us to sequence, just take a soil sample and identify bacteria based on the sequences of DNA that are extracted from that soil sample. Okay? Just to show you how fast this technology has advanced, the Human Genome Project was 20 years long, and about 80% of the human genome was sequenced in the last 2 years of the study. Think about that for a second. And the current round of gene deep sequencers are much faster than what was used there. So this opens up entirely new avenues to examine endophytic bacteria, which are very, very difficult to study. They don't culture well. Most of them you can't grow in a petri dish. Most of them, when you section a plant leaf and look at it under a microscope, you can't see anything. You can't see diagnostic structures that tell you this is what's going on the way you can with a root nodule. But these tools are letting us look at these things. I have 2 studies I wanted to share with you, and I'll put the titles and information that you can find these in the caption below this video. The first is titled Microbial Diversity of Paulownia Species Leaves, a New Source of Green Manure. Well, that should get your attention. We like green manure in permaculture. We like it a lot. So new sources are good. This study crushed up some leaves and looked at the full diversity of bacteria that were in them. And they discovered that 53%, almost 54%, were Hymenobacter, which is a nitrogen-fixing species. And in addition to that, almost 6.5% were Rhizobium in the leaves of these plants. Not in a root nodule, in the leaves. Still an endophytic nitrogen-fixing association, but one with no visually identifiable diagnostic characteristic. Okay? This is a 2018 study. Now, while I was doing some fact-checking for this video, and I was looking up this Hymenobacter, I found a study published 2 months ago, June 2022. The title is Unraveling Nitrogen-Fixing Potential of Endophytic Diazotrophs of Different Saccharum Species for Sustainable Sugarcane Growth. Saccharum being the Latin name for sugarcane. And just to kind of give you a key take-home from this study, it found Azobacter, remember those guys that live in the rhizosphere? In the tissue of sugarcane. It also found Azorhizobium in the tissue of sugarcane. Okay? in addition to the aforementioned bacteria. It has a list, a paragraph long, of Latin names for nitrogen-fixing bacteria that were found in the tissues of sugarcane. So this story, and that's only a 2-month-old story, what does that mean for the ecology of our general environment? We don't know yet. It's a 2-month-old study. Give it some time. We need to do more work on this. But the point I'm trying to make is there's more work to do. If you look back over the literature of looking at nitrogen fixers in their habitats, there's some, like crimson clover, that you can plant in a field and the next year measure a huge difference in residual nitrogen in the field with versus without. There's also a lot of ambiguous studies. Studies where people would look at, like locust trees, planted with other things, and look at nitrogen buildup in the soil and find no statistically significant results. Well, why? One answer is maybe, possibly, they're not as good a nitrogen fixer as we thought. Or maybe there's so much nitrogen fixation going on in the background that we can't tell the difference. There's also a lot of confusing literature on plants like these guys, like the non-nodulating legumes, like the Banksia trees from Australia, where you can look at the leaves and you can see the biochemical signatures of high nitrogen fixation. And the scientists would look at the roots and find no nodules and say, well, darn, that's confusing. But there's a lot more to endophytic activity than just root nodules. So the fact that we have these new tools opening up new avenues of research is going to help us explain some of these enigmatic observations. So the conclusion about how widely distributed is nitrogen fixation in plants is that we don't have the ability to conclude yet. There's a lot more out there. So stay tuned and pay attention to the research. And you'll find a lot more interesting things to include in your permaculture planting. So if you found this video interesting, if you're like this kind of content where we mix the scientific and the practical, please go ahead and give it a like so that other people can see it and YouTube will promote it. And subscribe to the channel because there's a lot more coming here at Old Ways Rising Farm.